Hello everyone, welcome to today's live broadcast, Fast, Simple, and Sensitive Electrochemical Biosensing using Screen-Printed Electrodes, a high-throughput solution. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, the leading social media site for science professionals, and sponsored by Metrome USA. Metrome is one of the world's most trusted manufacturers of high-precision instruments for chemical analysis. The company was founded in 1943 by engineer Berthold Schoner in Hedesau, Switzerland. Today, Metrome is present in more than 80 countries with its own subsidiaries and exclusive distributors. Headquartered in Hedesau, Switzerland, the Metrome Group also includes Metrome Applikon and Metrome Autolab, manufacturers of online analyzers and instruments for electrochemical research, respectively. For more information, please visit www.metrome.com. Before we start, there are a few instructions. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too, and you can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, just let us know by clicking on the support button at the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenters. We are proud to welcome Laura Fernández Llano, PhD, and Susana Campuzano, PhD. Dr. Fernández Llano is the product manager at Dropsense. She joined the company in 2008 after obtaining her European PhD. Dr. Fernández Llano was trained in the electroanalysis group of the University of Oviedo and performed research internships in the Turin University in Italy, developing molecular imprinted polymers for chromatographic analysis at the Karl Franzens University in Austria, and developing screen-printed electrodes and their application on biosensors development. With strong experience in analytical techniques, molecular imprinting synthesis, and characterization of materials, she has wide knowledge of sensors development using fixed-film hybrid technology in clinical analysis and according to ISO 13485 protocols. Dr. Campuzano is an assistant professor at the Analytical Chemistry Department of the Chemistry Faculty of the Universidad Complutense in Madrid in Spain. She received her PhD in analytical chemistry from the university in 2004. She belongs to the Electroanalysis and Electrochemical Biosensors Research Group headed by Professor Jose Manuel Pingaron. Dr. Campuzano worked as a Juan de la Cierva researcher from January 2008 to December 2009 in the Department of Molecular Microbiology and Infection Biology and at the Center for Biological Research, Spanish National Research Council, CIB-CSIC Madrid, and as a research scholar in the research group of Professor Jay Wang at the Department of Nanoengineering in the University of California, San Diego from January 2010 to July 2011. The speakers' complete bios are found on the LabRoots website. I will now turn it over to Dr. Fernández Llano for her presentation.
Hello everyone. I would like to thank you for attending this presentation and also thank Susana Campuzano for her presentation today, where she will focus on her research in the area of sensors and biosensors using Dropsense products. The purpose of this talk is to check the different alternatives available in the area of screen-printed electrodes as suitable platforms for sensor and biosensor development. We will talk about these different options, including multiplexed analysis that can be easily performed through these platforms. Afterwards, Susana Campuzano will present the research work of her group where drop-sense screen-printed electrodes are used as platforms for developing sensors for clinical analysis. Sensors or biosensors are devices that respond to a specific stimulus and provide a usable output. There is a continuous growth on this area as the aim is developing useful tools for daily life control and monitoring. Point-of-care diagnosis for quick and efficient medical support, security applications where a risk or emergency exists, like chemical or biological weapons, quality control, for example, in food industry, in order to avoid risk in the health and assure food quality and organoleptic properties, environmental control, or monitoring any industry processes. Electrochemical sensors are just a subclass of chemical sensors in which the electrode is a transducer and converts the chemical response into a signal that can be detected by electrical instruments. On the slide, you have an example of a system of electrodes made by screen printing technology. Instead of having a bulky cell, we have a planar design with 3 electrode systems printed over ceramic substrate. Working, counter, and reference electrodes, and also the electrochemical cell is clearly defined. The sensors and biosensors take the related advantage of nanoparticles, nanomaterials, and new technologies in order to develop quick and fast analytical methods, which are an alternative towards expensive and sophisticated laboratory instrumentation. Screen-printed electrodes are based on thick film hybrid technology. This technology consists in the deposition of inks over a ceramic or plastic material. The inks can be metallic, like silver, gold, platinum, and are dried in successive phases. The design is defined by a mesh that it can be easily changed. The advantage of screen-printed electrodes when compared with conventional ones are they are cheap to manufacture due to the technology employed that allows large-scale manufacturing. Their size and planar configuration simplifies working with them. Just a small drop of sample can be used to cover the 3 electrode system. This design is robust enough as we are avoiding systems where glass is used, for instance, reference electrode. Through appropriate modification and method development, electrochemical sensors give good sensitivity and selectivity in their measurements. Electrochemical sensors and the instrument to perform the measurements allow miniaturization, and therefore portable and wearable sensing devices have become a reality. As it was explained before, screen-printed electrodes are just transducer of the chemical event. The working electrode can be of different materials: carbon, gold, platinum, bismuth, ITO, p-dot for transparent electrodes, and also including electronic mediators. On the slide, we see pictures of the surface of carbon and gold working electrodes. These surfaces can be modified with different nanomaterials, like metal nanoparticles, graphene, carbon nanotubes. With these modifications, we are able to modify also the electrochemical response of these electrodes and enhance the response according to our needs. It is also possible to modify the electrode surface with different biomolecules like antibodies, enzymes, DNA strands, or also any other synthetic element. These receptors introduce their selectivity and affinity to the sensor, improving the analytical performance of these devices and their selectivity to the analyte of interest. Turning now to electrode configuration, the design can be modified and adapted to our research and application needs. For example, single electrochemical cells, where the 3-electrode system is placed in the middle or at the edge of the ceramic substrate, analysis on punctual sampling can be easily carried out. There are also available arrays of 8 or 96 electrochemical cells for simultaneous and multiple analysis. Processing up to 96 different samples or optimizing an electrochemical method could be achieved in short time with this kind of systems. Other options are electrodes integrated in flow cells. In this case, a plastic cover defines a channel and the electrochemical cell itself. This setup is suitable for a continuous monitoring of sample, for example, in a continuous flow or stream. To illustrate the different possible applications or uses of these formats, we will have a look at different applications with these designs. In food industry, a fast, simple, and low-price analysis is highly demanded, not only for quality control, but also for a proper control of fermentation processes. Electrochemical sensors for in situ ethanol detection and checking the alcohol content in beer or wine samples have been developed in combination with the well-known catalytic properties of platinum nanoparticles. Platinum nanoparticles are mixed with the sample, and after shaking, a sample drop about 50 microliters is placed over the screen-printed electrode. The electrochemical measurement gives an analytical signal that is correlated to the alcohol content of the sample. The total time of analysis takes less than 2 minutes and can be easily performed on final product, already bottled, or during the fermentation process. A portable reader like Dropstat, programmed to perform the electrochemical measurement and correlate the signal to the alcohol percentage, gives directly on its screen the analytical value and simplifies as much as possible the analytical method. This chart illustrates the percentage of alcohol measured in 10 different samples of wine. The results obtained are within the tolerance defined by the European Union regulations. This is an easy, quick, and simple method for checking the alcohol content in wines. Next is the sample of working under flow conditions with a dual graphene working electrode. Each electrode is modified with 2 different enzymes, allowing the detection of lactate and glucose respectively in each of these working electrodes. The experimental setup is really simple. The flow injection system consists on a peristaltic pump supplying continuously buffer solution through the electrochemical cell. The sample is directly ingested through a septum and the electrochemical detection is carried out. In the first graph, the successive injections of 5 millimolar lactate are shown. We obtain successive peaks corresponding to the analytical signal from lactate sensor, while no signal is obtained from glucose sensor. On the second graph, successive samples of 5 millimolar glucose are ingested. Only glucose sensor responds, while no signal is obtained from the lactate sensor. Finally, if we ingest a mixture of glucose and lactate, we obtain at the same time 2 analytical signals. This allows us a continuous monitoring of both analytes at the same time. The linear range for each molecule are large enough for real applications. To sum up, with this setup, simultaneous and continuous analysis can be easily performed. Moving on now to multi-well formats for high-throughput analytical systems. These kind of designs are very interesting for applications in the pharmaceutical and environmental or clinical fields. Parallel tests are run simultaneously with minimum operator intervention, increasing experimental load without subsequently increasing personnel cost or developing time. In this slide, we see the 96 well plate, a connector, an automatic controller connected to multi-channel drop sense STAT 8000 or single-channel drop sense STAT 400 instruments. This way, we synchronize the measurements in the wells. In each well, we can apply the same or different electrochemical technique. Therefore, we can design different assays according to our requirements. The versatile use of this system is shown in the following slides. Each well has at its bottom 3 electrode systems, a screen-printed working reference and counter electrode. Different materials can be used as working electrode: carbon, gold, platinum. As you already know, different modifications can be carried out on the electrode surface with carbon and metal nanoparticles, for example, in order to enhance the response. Therefore, at each column, we can run measurements applying the same technique for calibration or detection of the same analyte, and it's also possible to apply different techniques if we were analyzing different analytes in the same sample. The options are multiple, and this system is highly compatible not only with drop-sense multi-channel systems, but also with any multi-channel potentiostat such as Metron Autolab. To illustrate this, here is an example of cardiac biomarkers detection. On each well, an immunological reaction is carried out for the detection of cardiac troponin. An appropriate selection of detection antibodies that gives us selectivity to the analyte, and the electrochemical detection method, detection through alkaline phosphatase system, provides us sensibility in the detection. Applying solutions of no concentration of the analyte in different wells and running out the simultaneous electrochemical measurement of these different wells, we get the calibration plot. The correlation curve is shown in the slide with a limit of detection of 0.05 nanograms per milliliter. Once the method is optimized and the analytical characteristics are defined, as we have up to 96 wells where the detection can be performed, detection of these biomarker concentrations in different samples can be also carried out, allowing high-throughput sample detection. To summarize, this type of electrode format allows us to process multiple different samples. This reduces the time of analysis, an issue especially interesting if we consider routine analysis where a high number of samples must be processed, for instance, clinical analysis. Also, multiple analysis of the same sample can be carried out for the statistical treatment of the obtained data. Of course, calibration and analysis can be performed on the same well plate, and even we could have detection of different analytes on the same sample, just by developing different analytical methodologies on each well. Therefore, this system can be applied to a great variety of assays and formats, being very useful for these pharmaceutical, environmental, and many other applications. In conclusion, screen-printed electrodes are versatile platforms due to the different designs, electrode materials, and modifications. They have also reduced price and their distinctive disposable nature that avoids cleaning processes, making them very attractive for real applications. Moreover, the low volume of reactives needed reduces the cost of their modification, especially important when biomolecules are used. Finally, low volume of sample is needed, really critical when performing clinical analysis. In combination with portable instrumentation, point of care and in situ analysis can be easily performed. I would also like to recall that aside of our catalog screen-printed electrodes, we are also able to customize design according to your specific applications. Now it's time to give the floor to Susana Campuzano, who works in the group of one of the key leaders of electrochemistry research, Dr. José Pingarrón, group that have been for years performing many studies with our screen-printed electrodes and equipment. Thank you for your attention.
Thank you for that informative presentation, Dr. Fernández Llano. Yes, we will now hear from Dr. Campuzano. I will now turn the presentation over to her.
Thank you very much. Good morning to all. In the first place, I would like to thank Metron and DropSense for the opportunity to share our research experience. In this presentation, I will give, through selected approaches developed recently in our research group, a brief overview of the interesting possibilities offered by screen-printed electrodes in electrochemical biosensing of clinical relevant analytes. Nowadays, there is a huge demand for fast, reliable, and low-cost analytical systems for the detection and quantification of molecules with relevant in medicine, food, environmental pollutant monitoring, detection of foodborne pathogens, and bioterrorism. Indeed, the development of biosensors fulfilling the requirements of ultrasensitivity and selectivity, as well as convenience of use and cost, is one of the grand scientific, engineering, and educational challenges of the 21st century. In this context, electrochemical biosensing is considered as a particularly well-suited strategy for this purpose, as a consequence of its inherent high sensitivity, simplicity of the operational procedure, easy availability, and affordable cost of the required equipment, together with possibility of miniaturization and suitability for in-field applications. At present, the methodologies commonly used for determination of analytes of relevant in clinical field can be classified in techniques which detect the presence of target analytes by amplifying a fragment of a specific gene through the polymerase chain reaction, and assays which determine a specific protein or total protein. The main disadvantage of these conventional techniques are false positives, the difference between the results provided by different ELISA kits, the need to have a suitable standard for PCR, their challenging application in complex clinical matrices, and their limitation for use as point-of-care testing devices. In this sense, disposable electrochemical biosensors are emerging as powerful alternatives to these methods of determining analytes of protein and genetic nature, because they allow making determination quickly and easily with high sensitivity and selectivity, and using low-cost instrumentation, office automatization, and miniaturization, meeting the highest standard demanded by routine determination and decentralized analysis. On the other hand, magnetic particles are being widely used in the development of these biosensors because their use avoids laborious protocols of modification of the electrode substrates and improves their analytical performance in terms of sensitivity, test time, and minimization of interference by sample matrix. Screen-printed electrodes are recommended over other types of electrodes for this application, so they can be fabricated from several different materials, made in diverse geometries and multiplex formats, and offer the possibility for mass production at a low fabrication cost. In addition, the mass size of these electrodes enable a very small volume of sample to be used for analysis. Moreover, their planar shape facilitates the incorporation of magnetic bioconjugates by simple attraction using a magnet placed under the electrodes. During this webinar, I would like to discuss briefly some of the most relevant biosensing platforms developed in our research group for the individual or dual determination of relevant analytes at different molecular levels in complex clinical samples using screen-printed electroanalytical platforms. These developed bioplatforms are based both on the use of magnetic beads and integrated scaffold at plain and nanostructured screen-printed electrodes. Starting with the integrated bioplatforms, the first example I would like to talk to you about are disposable electrochemical DNA sensors for the detection of a target DNA sequence on the p53 tomo suppressor gene. These electrochemical platforms consist of screen-printed carbon electrodes, functionalized with a water-soluble reduced graphene oxide carboxymethylcellulose hybrid nanomaterial synthesized in our lab, and an amino-terminated terpene-specific capture probe covalently immobilized through carbonamide chemistry onto the nanostructure platforms. By using streptavidis peroxidase conjugate as an electrochemical indicator, the hybridization was monitored by recording the amperometric response measured upon the addition of 3,3,5,5 tetramethylbenzidine or TMB as a redox mediator and hydrogen peroxide as an inside substrate, leading to a typical on-off change as a consequence of the remarkable increased distance between the attached enzymatic conjugate and the electrode surface, which hinder the electron transfer. The synthetic target sequence used corresponds with a region of the wild-type gene and the single mismatch sequence with a mutant genotype that contains a single nucleotide change at codon 175, leading to cancer triggering the activation of this tumor suppressor protein. Under the optimized working condition, the analytical characteristics obtained for the synthetic target are summarized in the table. This platform allows a limit of detection of 2.9 femtomole and demonstrates a 15 days storage stability at 4 Celsius degrees. The selectivity results shows a complete discrimination between perfectly matched and the single base mismatched DNA. At this point, it is important to mention that since the TP53 gene shows little difference in expression between cancer and healthy tissues, it is much more relevant to implement a DNA sensing platform able to detect single mutation than variation in gene expression. Regarding the non-folding properties, results demonstrate that although with a lower sensitivity, it is possible to measure 100 nanomolar of the synthetic TP53 target with single nucleotide mismatch discrimination, both in spike-treated human serum and saliva samples, demonstrated that no significant absorption of the different matrix components occur on this novel scaffold. In order to demonstrate real clinical applicability, the implemented methodology was applied to analyze the endogenous TP53 status in different human cell lines. The used methodology implied the synthesis of a cyclic DNA by reverse transcriptase from previously extracted total RNA. The figure shows the results obtained for 3 breast cell lines, 1 epithelial non-tumorigenic and 2 cancer cell lines. Results provided were in agreement with the TP53 gene status reported in these 3 cell lines, thus demonstrating the sensor usefulness for this purpose. The second example I want to show you briefly relies on the development of a simple and cost-effective competitive RNA-RNA hybridization assay using nanostructured disposable electrodes. This work was made in collaboration with Professor Rouafi group from University of Tunis El Manar. The strategy involves a thiolated RNA complementary to the target microRNA assembled on a gold nanoparticles modified screen-printed electrodes and the competition with a biotinylated 4-stranded RNA with identical sequence to that of the target microRNA to be detected. The sequential assembly of mercaptohexanol ensured that the thiolated probe stand up and extend to the solution while minimizing non-specific absorption. The biotinylated microRNA competed with the target microRNA for hybridization with the thiolated RNA immobilized, and labeling was made with a streptavidin HRP polymer. The analytical readout was obtained again amperometrically, in this case with the hydrogen peroxide hydroquinone system. The higher the concentration of the target microRNA, the lower amperometric response was measured as a consequence of the smaller number of biotin microRNA molecules attached to the electrode. Under the optimized working condition, these scaffolds demonstrate a storage stability of 3 months, a limit of detection of 25 femtomolar, a linear dependence from 100 femtomolar to 25 picomolar between the diminution of the current value obtained compared with the current achieved in the absence of target microRNA and the target microRNA concentration. The selectivity evaluation demonstrates a complete discrimination between the fully matched and the single base mismatch sequence. This biosensor was applied to the determination of mature microRNA 21 in raw total RNA extracted from cancerous and epithelial non-tumorigenic breast cells. The results provided by this disposable platform showed substantially lower amperometric response for RNA extracted from the MFC7 cells, indicating upregulation of microRNA 21 in this cancer cell line. It is important to mention that accurate quantification could be accomplished simply by interpolation of the current difference measured for the total RNA extracted into the calibration graph constructed with target microRNA standard solution. Quantitative results achieved were in good agreement with data reported by other authors, thus demonstrating the feasibility of the developed approach to perform accurate determination of the target microRNA directly in total RNA extracted from cells using a much lower amount than required by other approaches. As an example, I have selected a very attractive disposable immunosensing platform for the simultaneous determination of two obesity-related hormones, ghrelin and peptide YY. In this case, dual screen-printed carbon electrodes were fairly modified with reduced graphene oxide, and after grafting of the diathonium salt of 4-aminobenzoic acid on this modified electrode surface, the corresponding capture antibody for its target hormone was covalently mobilized onto its working electrode using carbonamide succinimide chemistry. The determination of each hormone was performed by direct competitive immunoassays with the corresponding biotinylated hormones for the capture antibody immobilized. After labeling the attached biotinylated hormones with a commercial polymer of estrostabidine phosphatase, the DPB signal obtained in the presence of 1-naptyl phosphate was monitored. Under the optimized working condition, the analytical characteristic estimates summarized in the table were appropriate for the determination of both hormones at clinical levels in serum and saliva samples. Results obtained demonstrate also the reproducibility of the fabrication and functioning of the proposed immunosensing platform and their storage stability during at least 10 days. After demonstrating an excellent selectivity for both hormones against other proteins and the absence of cross-talking between the two target hormones and the two adjacent working electrodes, the applicability of this immunosensing platform to the analysis in spiked human serum and saliva samples was evaluated. Results achieved demonstrated that the accurate determination of both hormones could be performed simply by interpolation of the electrochemical signal obtained for sample diluted 1-1 into the respective calibration plot constructed with a standard solution. These results confirm the suitability of the approach for the analysis of these complex samples after minimal sample treatment. Regarding the coupling of screen-printed platforms with magnetic beads, as a for example, I would like to present you the most relevant results achieved in the development of an electrochemical bioplatform for the simultaneous determination of two analytes of different nature, the protein interleukin A and its associated messenger RNA, in undiluted saliva samples. Salivary levels of these two biomarkers have demonstrated to allow an accurate discrimination between oral cancer patients and healthy people. The implemented methodology involves the use of functionalized magnetic beads with specific antibodies against protein and a specific helping DNA proof for mRNA, and amperometric detection at disposable screen-printed dual carbon electrodes. In this case, the disposable electrode is placed in a homemade magnetic holding block with two neodymium magnets encapsulated to ensure the reproducible and stable magnetic capture of the modified magnetic beads onto its working electrode to perform the amperometric measurement. This methodology exhibits high selectivity and sensitivity for the target analytes, providing a detection limit of 0.21 nanomolar for mRNA and 72.4 picogram per milliliter for protein in undiluted saliva samples. It is worth to mention that the limit of detection achieved for the protein is 23 times lower than the clinical cutoff value established in saliva samples to discriminate oral cancer patients from healthy individuals. This dual amperometric biosensor was applied to the direct determination of both biomarkers in spiked raw saliva samples and to determine the endogenous content of the target protein in saliva samples from healthy individuals. It is important to mention that despite the heterogeneity and complexity of this biological sample, there were no statistical difference between the calibration plots constructed using saliva from 10 different healthy individuals, which imply the possibility of using a single calibration plot for all saliva samples analyzed. The results obtained in the analysis of spiked samples confirm the reliability of the methodology to determine low concentration of both biomarkers in raw saliva following a simple working protocol. Regarding the determination of the endogenous content of protein in saliva samples, the obtained results were statistically in agreement with those provided by a commercial ELISA kit and as expected from samples collected from healthy volunteers below the commonly accepted cutoff value. The last example I want to show you is a very recent strategy focused on liquid biopsy analysis involving the rapid determination of the humoral immune response in cancer patients using allotac fusion protein modified electrochemical bioplatforms. The immune system produces autoantibodies against altered tumor-associated antigens. The production of these autoantibodies may serve as indicator of tumor initiation, treatment response, or disease prognosis. Remarkable, the accurate detection of autoantibodies can be used for early diagnosis of cancer patients, since the humoral immune response in cancer appears several months or even years before the clinical symptoms, and due to the amplification by the immune system, autoantibodies are indicative of the presence of few tumor cells. Despite these interesting features for early cancer diagnosis, the fact that patients develop autoantibodies to multiple tumor-associated antigens is a major limitation to get clinically validated assays, because autoantibody signature diagnosis panel should be accurately defined. A likely candidate in such panel should be the protein p53, because serum p53 autoantibodies are reported in 10 to 40% for all cancer patients. The developed electrochemical biosensor combines for the first time the use of magnetic beads modified by covalent immobilization of C-terminal Taq allotan fusion in vitro express proteins as efficient magnetic microcarriers to selectively capture the autoantibodies against the immobilized protein. The developed approach substitutes the use of purified expensive cancer proteins for allotaq fusion protein produced just in time for the assay using a cell-free system, abrogating potential concerns about the stability and integrity of the protein during storage. The fundamentals of the first electrochemical biosensor described so far for the quantification of p53 specific autoantibodies in sera and the reaction involved in the electrochemical transduction are displayed in this figure. Upon capture of the autoantibodies against p53 in vitro express protein, the attached autoantibodies are detected with a secondary HRP conjugated anti-human IgG. Then, the magnetic beads bearing the immunocomplex are magnetically captured on a screen-printed electrodes, and the biorecognition event is monitored by amperometric with the hydrogen peroxide hydroquinone system. The detection of the presence of autoantibodies to p53 was carried out on a non-characterized population of 62 sera from individuals at the high-risk program to develop colorectal cancer. ELISA test determined that 32 out of the 62 analyzed sera were positive for the presence of p53 autoantibodies, with an absorbance 5 times higher than the negative control. The seroreactivity to ERBNA1 protein employed as a control confirmed the selective seroreactive to p53 in the analyzed population. Under the selected experimental condition, the calibration plot, constructed with a commercial standard, demonstrates that the sensitivity of the biosensor is 440 times higher than the ELISA methodology, which allows a better discrimination between p53 reactive and non-reactive serum samples. The diagnosis effectiveness of this biosensor was evaluated by analyzing 24 human sera, and the results were compared with those obtained by using the ELISA test. In general, both methodologies detected higher levels of autoantibodies in p53 seroreactive patients, although the remarkable lower sensitivity of the ELISA kit made the conventional methodology more prone to produce false positive results. It is important to remark that the mean value of the signal obtained with this methodology for p53 seroreactive and non-seroreactive patients confirmed that a better discrimination between both types of samples was achieved with the electrochemical biosensor in comparison with the commercial ELISA test. Additional data were obtained in the analysis of sera samples collected from patients already diagnosed with cancer, 4 colorectal and 2 ovarian. The results are summarized in this table, and in all cases are in good agreement with the cutoff value of 120 units per milliliter established by the commercial ELISA kit to discriminate between positive and negative samples for p53 autoantibodies. These results confirm again the reliability of the developed electrochemical biosensor, also to perform selective determination of the p53 autoantibodies and sera samples from different cohorts of patients. The versatility and selectivity of the methodology was demonstrated by immobilizing different allotaq fusion proteins to magnetic beads. Since these results demonstrate the possibility of easily translating this methodology to the determination of autoantibodies specific to other proteins apart from p53 without particular parameters optimization, currently work is ongoing to develop a platform for the screening of humoral immune response in cancer patients through multiplex electrochemical determination of autoantibodies against different tumor-associated antigens. These results demonstrated that the implemented electrochemical sensors, based on the convenient use of commercial screen-printed platforms, can be employed as a user-friendly, cost-effective on-site device operated at room temperature for the selective and sensitive single or multiplex determination of target analytes of the same or different molecular level in complex clinical samples without prior amplification, preconcentration, purification, or labeling step, ideal for point-of-care application. This novel electrochemical biosensing approach compares advantageously with other analytical methodologies available in terms of simplicity, rapidity, portability, and cost affordability, and therefore can be easily transferred to the productive sector to develop easy-to-use and portable device for point-of-care purpose. It is important to mention also that although the concept has been illustrated for the determination of clinical relevant biomarkers, the developed strategies could be readily translated to the determination of other target analytes and find application in other interesting research fields, such as food quality control. I hope this presentation has given you an overview of the tremendous capabilities and great versatility currently offered by commercial screen-printed platforms for the individual or simultaneous determination of analytes of the same or different molecular level, both using integrated formats or coupling with magnetic beads. Finally, I would like to thank all the people and the financial funding that have made possible this research, to the great research teams led by Professor Pingarron at UCM and Professor Joseph Wan at UCSD, with which I am fortunate to continue working, to Metron, Rosen, and Laura, and especially to all the people registered to this webinar. Thank you very much to all of you for your kind attention.
Excellent presentation, Dr. Campuzano. Thanks to both speakers for bringing that information to us. So before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. And our first question is for Laura, and it is, why a ceramic substrate? Plastic works just as well, assuming no exposure to organics won't degrade plastic.
From our experience and due to the surface characteristics of the ceramic, what we have seen is that the ceramic allows the use of more different inks, more different modifications, and also the designs that you can lower the dimensions of the electrodes in a very good way over ceramic. This doesn't happen with plastic, so that's why we usually use ceramic, but of course both of them can be used.
Okay, this next one's for you too. What's typical in the enhancement of signal strength when using any of the classes of sensors described?
This enhancement of the signal depends on the method or the modifications that you perform on the analyte. So there is not a value, and should be checked with the molecules, with the target molecules.
Okay, this next one's for you too. Is there any method to reuse these screen-printed electrodes?
This is a very typical question. And of course, they are supposed to be single-use, but depending on the method and the analyte, sometimes they can be reused, just by applying some electrochemical treatment, or for example, in some of the applications that we have seen, we are using the same electrode for successive analysis. So it depends a little bit on the analytical method on the analyte. So we can say that can be also reused.
And Laura, for alcohol, do you use a calibration standard before you use the SPE unit?
For this specific application, what we have done is a calibration plot that it's introduced the trough that this device is programmed with this calibration plot, and we can correlate the signal that we have in our sample to the signal that we obtain with the signal obtained in this calibration plot. Of course, in order to validate this protocol, we should use a standard concentration of alcohol that has been verified by other analytical methods.
And Laura, how do you assess the incorporation of the gold or graphene nanomaterials on the surface of the disposable electrode?
This is a very nice question, and the way of checking this, it can be by techniques like scanning electron microscope, so we can see the surface of the working electrode, but also by electrochemical techniques that we can detect the amount of gold that is deposited over the working electrode. These are just some examples. Mm-hmm.
Next question is for Susana. It has several parts. Let me know if you need me to reread any part. How does the dual working electrode containing carbon electrodes work? Does both working electrodes operate by single reference and counter electrodes, and can they be used for simultaneous detection of two analytes?
Yes. In that, we use the same counter and reference electrode for simultaneous detection of two analytes. Yes.
And did you need me to, did that answer the whole question? Did you need me to repeat any part of it?
Sorry?
Oh, did that answer all parts of the question? Do you need me to repeat any part of the question, or that covered it?
No.
Okay. We can move on to the next one, which is for both of you. This person says, sorry, I didn't get it. Does the multiple well sensor system include a magnet below each well to work with magnetic beads? So Laura, do you want to take that first, and then Susana?
It's because I cannot hear you properly, sorry.
Shall I repeat it?
The sound is good now, and I cannot hear properly the question. Can you repeat again, please?
Oh, yes. The person says, sorry, I didn't get it. Does the multiple well sensor system include a magnet below each well to work with magnetic beads? And again, this one is for both Laura and Susana.
Yeah, I mean, the 96 multiplex includes the magnet below. You can place this part up, and you can use this format with magnetic beads. This is the things that we are working now on our lab. Yeah.
Okay. And Susana, did you answer that one too?
Okay. Laura. I don't.
Yes. Laura?
No. I mean, I agree with Susana that it's the way to work with these systems, placing a magnet below the working electrode, also in multi-well sensors.
Yeah.
Okay, and the next one is for Susana. How do you avoid the lines of the magnetic field from congregating in small points on the working electrode, thereby limiting sensitivity?
Okay, I think we have not seen any effect of the magnetic field because we usually, I mean, with the template that DropSense use for printing the electrodes, the distance, this small distance is more than enough to avoid the electric field or the small neodymium magnet that we use below the electrode to capture the magnetic bead. We have not seen any effect or any interference from the electric field in our electrical measurement.
Okay, and this next one is for Susana. Did you ever observe any absorption of interference on electrode surface, and if yes, how do you handle or prevent it?
I mean, we normally use the usual blocking solution containing BSA, casein, or even power milk, and it should, or ethanol, I mean, it should be more than enough to block the non-specific absorption. There are many commercial buffers that are specialized for this purpose that they work really good. And also we work a lot on the surface chemistry, putting mercaptohexanol and different modifications to about the non-falling properties. It depends on which type of format we are using. I mean, for the work with magnetic beads, the minimization of the non-specific absorption are performed with a buffer solution, and in the case of integrate electrode, we work on the surface, putting non-falling agents like mercaptohexanol, ditiols. That's it.
Okay, and Susana, can this biosensor test whole blood?
Whole blood?
Yes. Yes.
We have not tried yet whole blood. We have tried raw serum and raw lysates, and it works, and raw saliva, and this is what very good. We have not tried raw blood, but we will try in the next future for sure. But I hope no problems. We have not tried for the moment raw blood because it has hemoglobin, and hemoglobin is a peroxidase, and the peroxidase is going to give an interference in our electrochemical signal. But this is the only reason. But if we change the enzymatic marker and we use phosphatase alkaline, for sure we are not going to have problems with blood.
Okay. And Susana, are the measurements a transient value or terminal value? In other words, glucose meters take transient measurements.
There are terminal values.
Okay. And the next one is for both of you. What is the electrode to electrode variation in a multi-electrode array?
In a screen printed electrode? Sorry, Susana.
I mean, for our expertise, the reproducibility between the electrodes is very, very good, both in the integrate formats and in using only plain for as detectors. We always got reproduce or RCD values below between 5 and 10%, and we are making many different steps. Then the reproducibility and the fabrication is very, very good.
Okay. Thank you, Susana, because because you were asking about that, but yeah, it's a high reproducibility product, so the electrode. Yeah,
I cannot present this kind of application with not reproducible electrodes. It's impossible.
Okay, this next one is for Susana. In the gene TP53 gene detection, what type of hybridization and detection buffers were used?
Okay, for the hybridization, we use the conventional PBS buffer, phosphate saline buffer. And for the detection, we always use the same buffer, only phosphate buffer at pH 6, 0.05 molar. And this is the thing that the buffers that work better in this case.
Okay, we're going to pause 1 second to see if more questions are coming in. So let's just take a quick pause and we'll resume in 1 quick second. You know what, we've taken a lot of questions today. I think we'll stop now and we'll give the speakers a chance to give a last word. So I would like to once again thank Dr. Fernández Llano and Dr. Campuzano for their presentations. And would you like to share some final comments with us?
Yes, thanks again everyone that was attending to this presentation. Thanks Susana for her fantastic work with these different screen printed electrodes, and I'm quite sure that the applications that they have developed will come in a reality for clinical analysis soon. And just let you know that we are open for more questions. Just contact Metron USA or Dropsense directly. I think also Susana will be able to answer your questions and requests. That's all. Thank you.
I would like also to give thank you to all and to Dropsense and Metron for inviting me. It has been really a pleasure for me speaking about this work that we cannot make for sure without good tools to develop our assays, and that's all. Thank you all.
Can I just ask you too, I thank you for that. Another question has come in, and if you're able to take it, that would be great. Can the blocking layer solution be impregnated on a solid membrane? Are we able to take that one today, or should we wrap up?
Laura, can you reply them, or?
Sorry, because the blocking layer solution, what does mean?
Oh, I can just repeat the question, see if just can the blocking layer solution be impregnated on a solid membrane?
In the membrane of, I suppose in the membrane, I don't know, I don't understand which membrane I'm referring to.
Okay, well maybe after the event is over, you can speak with the person asking the question,
okay,
and
better,
come up with a firm answer for that. Okay, well thank you again. I would also like to thank our sponsor, Metron, for making it possible to bring this presentation to you. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Today's webcast will be available for on-demand viewing through July 2017. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You are welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. We'll see you next time.